Thank you very much for the invitation to speak here. I'm probably not going to stick to my abstract quite, but I'm going to spend a little bit more time talking about methodology, just because I think it's actually important to get a feeling of the range of things you can do using genome editing technology. Obviously we know, and we had a talk, very nice talk this morning, about the, genome editing can be used, tools can be used for all sorts of things in basic research, and that's really the huge excitement around the development of these methods, particularly CRISPR-Cas9 methods, is their importance in aspects of basic research. We had all these lovely talks this morning about the ability to read DNA and find out which genes are perhaps involved in which clinical syndrome. But these tools provided by genome editing have the prospect of rewriting DNA and to maybe correct some of the diseases. So that's really what I've been asked to talk about. And I'm going to go through, not going to really talk about basic understanding of human biology, but of course it's really important in any aspect of human biology. You can probably use these techniques to study. Create models of human disease, again, very important. But I'm really going to focus my talk really on somatic gene therapy, and then the possibility of using germline changes to avoid or prevent genetic disease. And I'll touch towards the end on possibilities of extending this beyond what people are mostly comfortable with in terms of enhancement. Okay. So in terms of respect to somatic gene therapy, genome editing is, of course, a relatively new tool for gene therapy. Traditional gene therapy, as you will know and you will hear subsequently, involves using a viral vector to integrate one or more copies of a functional version of a gene basically at random in the genome. Hope that it's going to be expressed in the right place at the right time and at levels which are going to have therapeutic benefit. So there's some random aspect to all of that, of course. And then the vector can also, because it integrates, it could disrupt a critical gene when it does so. So there has always been concerns of what happens if you mutate, for example, lead expression of an oncogene, or you mutate a tumor suppressor gene, for example. So genome editing clearly provides a way to make desired changes in the gene of interest, which could be to correct a gene causing variant. The gene will hopefully retain its normal expression pattern, and other genes should not be affected if it all goes to plan. Traditional gene therapy may also not be of so much use if a disease is due to the expression of an abnormal protein, because you will still always have that background of high expression of the abnormal protein. So genome editing gets around that problem as well. So in terms of somatic gene therapy, there are two broad approaches that people think about. There are ex vivo approaches where you remove cell population, and the obvious ones are bone marrow stem cells for treating disorders that could be, for example, sickle cell disease or thalassemia. Of course, you can also do treatments such as use CAR T cells to try and treat cancer. There are possibilities of using other stem cells, which you could take out of the body, grow them, manipulate them in vitro, and then put them back. Ex vivo approaches are very attractive because you can verify that you've got the correct genetic alteration that you want and no other major problems in the cells before you put them back in the patient. But of course, that also adds to their cost, because to do this in a way that's safe is really expensive. You could also contemplate doing in vivo somatic gene therapy, where you have to somehow get your gene therapy components, so your CRISPR-Cas9, etc., genome editing components, for example, into the body efficiently enough. And so this generally relies on using things like a viral vector. There are other methods that will work for some organs, like nanoparticles. There are a number of examples, and I'm not going to spend any time on talking about the examples, but there are examples which are already going to the clinic, for example, editing retinal cells, liver cells for metabolic diseases, muscle cells for muscular dystrophy, etc. So if I'm talking, just going back to basics a bit about genome editing and the methodology, so it most often requires a nuclease enzyme that will make often a double-stranded cut in the DNA, or in some methods just a single-stranded cut or nick in the DNA. You need a mechanism to recognize specific DNA sequences. These can be derived from DNA binding proteins, such as transcription factors in the case of zinc finger nucleases and talins, or complementary RNA, which is the CRISPR-Cas9 methods. And if the aim is to make more than a simple mutation, you need also to have something like a DNA template with homology arms that's going to allow homology-directed repair. There are some other methods which I will mention which are actually looking very promising as well. You can also have modified forms of the nuclease, for example, the Cas9 nuclease, to actually alter the activity of genes rather than cutting DNA. Which is something that we need to keep in mind when we're talking about using genome editing approaches to treat diseases. So the three main methods that have been all entered or entering the clinic are based on zinc finger nucleases. I'm not going to spend any time on these talins. So these both rely on these transcription factor-like domains to give you your target specificity. FOC1 as your nuclease, you need two FOC1s to make a double-stranded break in DNA, which is why you have two sets in each of these cases. That helps specificity, but it becomes a lot more cumbersome to make and use. CRISPR-Cas9 uses the guide RNA to take you to the right place in the genome, and then Cas9 is the nuclease. I'm going to talk mostly about CRISPR-Cas9 because that's where the activity is in terms of new research, but mustn't forget the other ones which are also very useful. So there are now many CRISPR-affected nucleases that can be used, which vary in their properties in terms of requirements for specific sequences to allow them to interact properly with the target DNA, the so-called PAM sequences. Size, where you could use a small one like this one much more readily in a viral vector because there's room for it. They also vary in terms of on-target versus off-target specificity. So, for example, HyperCas9, published by Jennifer Doudna 2 years ago, definitely is much more accurate. So it tends to hit the on-target site much more than any off-target site. So just very briefly, and don't need to focus on the details, but if you can use CRISPR-Cas9 just to make an inactivating mutation, as we heard this morning. All these methods rely on DNA repair mechanisms, and in this case, it's relying mostly on non-homology end joining or microhomology-mediated end joining. So you make a double-stranded cut in your DNA, and the repair process is inaccurate, often leading to a small deletion or insertion at that breakpoint, relying on these two methods. So this is a really quite an efficient way of making a mutation, either within a coding region of a gene, or you could target an enhancer, for example. It can also be, of course, useful for certain types of gene therapy. For example, Duchenne muscular dystrophy, it's proposed to use this method to promote exon skipping. So you skip the exon where the mutation is, but enough of the protein can still be made to give you a functional protein and relatively normal muscle function. You can try and have homology-directed repair. So this is using CRISPR-Cas9, for example, to exchange sequences. So you're substituting one sequence for another. In this case, you start off in the same way, so Cas9 protein guide RNA, but you also introduce a DNA template which carries the particular change that you want to make in the genome. I just illustrated an 8 base pair substitution here, but you can really alter anything from a single base pair up to many, many kilobases. You can make insertions or deletions or substitutions. Okay. There are many, many uses of homology-directed repair, of course. So you can alter coding region of genes, you can alter splicing, you can alter regulatory regions of genes, insert marker genes, insert bioactive genes from different species. You can, whatever you can imagine, in theory, you could do with this. But there's a problem in that homology-directed repair tends to only occur during S phase and G2 of phase G2 of the cell cycle. In other words, the cells really have to be dividing for this to work at all efficiently. And the other methods, particularly non-homology end joining, are very active at all phases of the cell cycle. So quite often when you're using this, trying to get homology-directed repair in the lab, you end up with quite a lot of instances where you also have non-homology end joining events. And so this is a problem with incorrect on-target changes to the genome. You have to be very aware of these. Every time you make a double-stranded cut in DNA, you may also get some inappropriate non-homology end joining event. Now, to get around this, David Liu and colleagues have invented a couple of techniques. So one of these is called base editing. In this case, you've inactivated the Cas9, so it doesn't make a double-stranded cut in the DNA, but instead it nicks it, and you make use of cytosine deaminase basically to alter a CG eventually to TA. So the C is chemically modified to become a U, no double-stranded breaking in DNA. The U then gets substituted with a T by endogenous processes, and then DNA mismatch repair detects a problem, repairs this, and you often get repair going this way, so you've substituted. They've been clever. They've also invented a new method to also go from AT to GC. So about many cases of inherited diseases are due to these sorts of single base pair changes in DNA. So these base editing methods become potentially very important to be able to deal with many cases of inherited disease. Okay. Unfortunately, base editing will not work for some common diseases. And I'm going to, actually, in my talk, use sickle cell disease as an example. So in this case, the single nucleotide change is where you have a GAG codon going to GTG. And you cannot use this base editing method for this. But David Liu last year came up with what looks like a very interesting method, which he refers to as prime editing. So you don't make a double strand break in the DNA. It uses a modified reverse transcriptase linked to both a dead Cas9 and the nickase, and then a modified guide RNA that both specifies the target site and encodes the desired edit. Now, so these guide RNAs include an RNA primer sequence, hence the name prime editing, adjacent to the guide RNA recognition site. This will then prime the reverse transcriptase, and then you have this template sequence on the RNA, which is when gets copied into DNA and substituted into the target sequence. So in proof of principle experiments, in completely the wrong cell type, they showed that it can efficiently convert the sickle cell mutation to wild type up to about 50% so really can be really efficient. Now, there's only still really this one paper published. We need to know a lot more about this method. Having reverse transcriptase active in the cell might have other consequences, so we have to have a lot more work done to see whether this could be safe to use. But it's very promising. I'm going to skip that just to say that you can use this to make any base change you desire, one to another. You can use it to make small insertions or small deletions or substitutions. As I mentioned, you can also use these methods to alter gene expression. I'm not going to go into this, but you should always just think about this. You can have a transcriptional repressor attached to your dead Cas9, or a transcriptional activator, or some chromatin modifying factor to alter gene expression. You have to think, though, that these effects may depend on the continued presence of the Cas9 and the guide RNA. Although with some changes you can imagine, they could be long-lived changes. All you need to do is switch at one point during development, for example, and then it maintains that pattern gene expression. So these are interesting to think about. It's a bad idea to have the Cas9 and the guide RNA present in the cell that you want to modify for very long. The longer it's present and the higher the amounts, the more likely you are to have incorrect on-target effects and particularly off-target events. So generally you want to keep it as short as possible. So there are more and more examples now of somatic gene therapy in humans using genome editing, and these are some of them. There are so in all these cases, the genome editing has been carried out ex vivo, but there are some trials beginning where it's in vivo. And Leber congenital amaurosis is one that has begun, and I think there's one patient, maybe two, treated, looking good, but very, very early days. To do your genome editing in vivo is much more challenging. We will hear more about somatic gene therapy approaches in vivo. So a good example of this would be muscular dystrophy. So there have been experiments conducted in animal models, particularly mice, but also in dogs, which look really, really very promising. But you have to use an enormous amount of viral vector to do this, and you have to always think, what are the likely problems with this? Just to, again, mention as an example, sickle cell disease. So what are the current strategies for dealing with patients with sickle cell disease? So there are drugs that relieve the symptoms, and actually, if you can give to newborn children, well, penicillin, followed by hydroxyurea, then they can survive reasonably well, and it can have big effects on survival. But it doesn't mean that they are always happy. In fact, they are going to grow up to be adults who can have sickle cell crises and suffer a lot. So, of course, people are thinking of ways of dealing with sickle cell disease. So there are a number of approaches that are entering trials. Of course, you can use bone marrow transplants from siblings, and that provides a long-term treatment, but bone marrow transplants are clearly not available for many, many people. So there's a suggestion that you could use CRISPR-based genome editing ex vivo. So one set of methods is based on this knowledge that persistence of fetal globin, so the gamma globins, which are usually turned off shortly after birth, can ameliorate sickle cell disease. So if you have persistence of those globins. And it's known that mutations just upstream of the gamma globin genes can reduce the binding of two transcriptional repressors, LRF and BCL2A. And if you have these mutations, it can inhibit the silencing of gamma globin. So you can use CRISPR to either boost gamma globin expression by mutating a specific enhancer of BCL2A, and that will reduce the levels of that transcriptional repressor. And this is the basis of a clinical trial that's already started. the first two people whose genes were edited have fared well. I don't know what that means exactly, but that's all I could find out about it. The other approach, of course, is to mutate the repressor binding sites upstream of the gamma globin genes themselves. And that's an approach that's been looked at in a number of experiments in animals, cell lines in animals. And it might work, but you may have to mutate both the LRF and the BCL2 sites. But you can't do that easily at the same locus, so you probably would have to use something like base or prime editing to get that to work. And then, of course, the other approach would be actually to correct the new mutation in the beta-globin gene itself, and that approach is being pursued very actively by several groups of scientists. And the efficiency so far is fairly reasonable in these experiments. So up to about 25% in one set of experiments look very promising, with a significant reduction in sickling. But actually, there are some suggestions that you can get even much, much higher frequencies if you play around with the techniques. So these look promising. But the problem, let me go back. One of the problems, of course, with tackling something like sickle cell disease, it's perhaps all very good thinking about doing this in a relatively rich country like the UK or the US, where sickle cell disease is an important problem. But how would you use this method? But every time you do this, it's going to be very expensive. So how would you deal with this problem in parts of Africa where the carrier rates can be up to 40%? 2% of children, I think, in Nigeria are born with homozygous for sickle cell disease. So the number of people with the disease is so much more prevalent, and of course, they don't have the money and the resources to do this. So how do you do this? You need to think of scalable methods that can be applied to not just a few very rich people, but to many millions of people. And that's a problem with somatic gene therapy, but of course it's also an issue with germline gene therapy, but we should think about that too. So what are the ways to allow individuals to have genetic-related children without passing on a known risk of genetic disease? So frequently, when you talk about the possibility of doing germline genome editing, people say, well, you have pre-implantation genetic diagnosis. Why can't we use that? Well, of course you can. But the methods are unacceptable for some individuals. Of course, they always put a burden on women who have to undergo IVF. And there are cases where they can't be used to retain a parental connection, genetic connection, but avoid the genetic disease. And I'll touch upon those in a second. Also, I'm going to mention this again, but I want to plant this thought in your heads. Consider patients who survive to breeding ages because conventional treatments or somatic gene therapy has now allowed them to get to reproductive ages and they want children. Are they going to want to have some difficult, maybe somatic gene therapy or other treatment later in life? Do they want their children to have to undergo all these procedures? No, they're going to want to have their germline corrected. So just think about that. PG is not always possible. So there are rare individuals who are homozygous for dominant versions of genetic disease, such as Huntington's disease. We'll hear about it. There are rare occasions where both parents are homozygous for recessive mutations. And in countries like Nigeria, where sickle cell is so rampant, you can imagine that happens quite a lot. Where mutations affect fertility can be an issue. For savior siblings, or where you have more than one harmful mutation that you need to correct at the same time, look at the same time, so with PGD, it often cannot work. You don't have enough embryos to find one that's going to give you a baby free from the disease. And then the genome editing methods may turn out to be more efficient. We don't know, not yet. Certainly not yet, but maybe more efficient. So one meta-study using PGD only 95 couples out of 358 brought home a baby. So that's a huge failure rate for PGD. Okay. And in cases of Savior siblings, this is an example provided by George Daly a couple of years ago. Harvard Medical School, 8 families involved in trying to find an embryo that was going to allow their first child to be cured of, in this case, X-linked immune deficiency diseases. 8 families, about 5 cycles of IVF each family, only 1 baby born from all 8. So only 1 family was able to take home a baby that could be used as a Savior sibling. This is very, very inefficient. So 2 main methods are potentially for heritable genome editing. So one is to edit cells that will give rise to sperm, such as spermatogonial stem cells. Okay, I guess that means I'm running out of time. I'll be very quick. So the advantage of going to precursors of the gametes is that you can verify the edits before any embryos are made. And this has been done in animal models, including macaques. But we are still quite some way off doing this in humans. So the other main route is by editing the fertilized egg. And we have a big problem here in it's going to be much more difficult to verify whether you have the correct edit and no other edits that you don't want. Unless it's known that the method is about 100% reliable, you can't rely on something like pre-implantation genetic diagnosis because you don't know which cell you're taking from the embryo. Is it one that's got the correct edits or not? So there's a huge problem. But the methods are becoming more and more efficient. All 3 homology-directed repair, non-homology end joining, and base editing methods have all been used in early human embryos. The famous case now of the 3 girls born in China who were modified in the CCR5 gene, that was just using non-homology end joining, which is the worst possible technique you might want to use to make a precise edit. So there's a lot of research directed at trying to increase efficiencies, avoid off-target and other unwanted effects. Methods of choosing when and how to introduce the genome editing. All of these are important, as are exploring these base editing and prime editing methods that avoid making double-strand breaks in DNA. Hopefully give you more accurate editing. I'm going to skip that. There are also methods, by the way, that you can try and do this in vivo. You could mention spermatogonial stem cells. So you could inject the genome editing components into the testis. Don't say ow too loudly. You could do that, and you might get enough spermatogonial stem cells to be able to have some chance of passing it on to your offspring. But you can also inject the components into the oviduct and hit the embryo. This is done with electroporation or using viral vectors. In utero. And you can get genome editing to work in both these cases. So then just finishing off, but which gene variants would you go for? Which are most obvious to think of? Well, I'm going to do this germline genome editing. It's difficult to focus on specific genes. You've got common diseases, such as cystic fibrosis, Duchenne's, sickle cell disease, etc. You've also got rare diseases, but the rare diseases can occur at very high frequencies in specific populations, so don't forget about those. So I would say this, perhaps it depends really on what the patients want, not what we as scientists and clinicians think would be the most appropriate. And then this point again. And then I said I'd finish briefly on enhancement. Let's not go into this, but a lot of these things you absolutely can't do, and we will probably never be able to do them, so we shouldn't even think about this. So as we heard this morning, how many genes are involved in height, be even more involved in intelligence. You cannot do this in any efficient way to design your baby for many of the traits that you might want your baby to have. The sorts of things that might be more interesting are ones that go outside the realm of normal thinking about disease, and things like diet. So, what if we could eat plastic? We could solve lots of problems at the same time. So I will stop there. Thanks.